Welcome to the Deep Dive. Today our mission is to, well, fundamentally rethink how your body knows the difference between a healthy part of you and a dangerous invader.
Yeah, it really is a complete paradigm shift we are talking about today.
It absolutely is.
Wow.
We are analyzing the findings of a truly groundbreaking Nature paper. This was published in April 2026 by researchers Fan, Crummell, and their incredible team.
And it is a fascinating piece of work.
Right, because they basically managed to catch our immune cells on camera performing a previously undiscovered microscopic theft.
Microscopic, that's exactly what it looks like, yeah.
But before we get into the lab work and the lasers and all of that, I want you to imagine your immune system as a highly trained, completely uncompromising bouncer at the most exclusive club in the world. And, well, that club is your body.
Right, and it is a massive security operation. You have, trillions of cells in that club.
Literally trillions.
Exactly. And the immune system has to constantly scan the crowd to ensure nobody is sneaking in a weapon or turning violent.
For decades, the fundamental understanding in biology has been that this bouncer learns to recognize the VIPs, like your healthy, normal cells, by checking the ID cards of guests who have, well, already died.
Yeah, the scavenging model.
Right. The prevailing theory was that the bouncer was basically a passive garbage collector, sorting through the trash at the end of the night to figure out who was supposed to be in the club.
And the scientific community has heavily relied on that scavenging model. The textbook understanding of how our immune system learns self-tolerance, meaning, how it learns not to attack your own body, assumed that specialized immune cells would just clean up the dead or dying cellular debris.
Like a cleanup crew.
Exactly. They read the molecular signatures inside that debris and catalog it. They would essentially say, okay, I found this ID card in the recycling bin. This is what a normal liver cell looks like. Let's make sure the rest of the immune army doesn't attack the living ones.
But today, we are completely flipping that script. This recent Nature paper has captured something on camera that changes everything. Our bouncer isn't just checking the IDs of the dead.
Not at all.
It turns out this bouncer is actively walking through the club, gently plucking tiny hairs off of the living guests to check their DNA, all without harming them. Okay, let's unpack this. Because to understand how revolutionary this active microscopic theft is, we need to dive into the incredibly dense biological mechanisms at play.
We really do. It requires a total shift in how we view immune surveillance. So, to set the stage for you, we have to look closely at a concept called antigen presentation.
Right, the border patrol.
Exactly. You have these specialized border patrol cells called antigen-presenting cells, or APCs. And macrophages are the really heavy hitters in this category. Their entire biological purpose is to constantly survey your tissues, gobble up proteins, and present those proteins to your T cells.
The T cells being the actual foot soldiers, right, like the ones that carry out the attacks.
Precisely. The macrophage holds up a protein fragment and essentially trains the T cell. It says, this protein is self, it belongs to us, stand down.
And if that goes wrong?
Well, if this training process fails, or if the macrophage presents the wrong information, you end up with an autoimmune disease. That is where the immune system's foot soldiers start actively destroying your own healthy tissue.
Wow. And the old dogma told us that these macrophages primarily gathered these self-proteins through a process called apoptosis.
Right, which is programmed cell death.
So a skin cell reaches the end of its natural life cycle. It neatly packages itself up for disposal so it doesn't cause inflammation, and then the macrophage eats those neat little packages.
Which, makes logical sense. But Fan and Crummell suspected that waiting around for cells to die just wasn't the whole story.
Because it's too slow, maybe.
Exactly. To test this, they engineered an incredibly elegant experiment using fluorescent proteins, specifically the Z's green protein. They genetically tweaked mice so that specific, perfectly healthy tissues would glow bright green under a microscope, basically like a neon sign.
But rather than making the whole mouse glow, they used these specialized genetic switches to target specific rooms in the house, right?
They did.
Yeah.
They utilized specific genetic promoters to isolate different tissue turnover rates.
Break that down for us.
Sure. So, for instance, they used a promoter called SCBG1A1 CRE-ERT2. I know, it is a total alphabet soup.
Yeah, that is a mouthful.
It is. But essentially, they engineered it so that only the slow-growing cells lining the airways of the lungs would glow green. These airway epithelial cells have a very long lifespan. They aren't dying and replacing themselves very often at all.
Okay, so that's the slow turnover environment.
Right. And then they also created a separate group of mice using a different promoter, K14-KER, where only the fast-growing skin cells glowed green, just to compare different environments.
So we have these mice walking around with neon green lungs or neon green skin. The researchers then isolated the white blood cells, specifically the macrophages patrolling those green areas, to see what they were carrying.
And they found something really strange.
Yeah, the macrophages were absolutely packed with tiny submicrometre-sized green dots. The researchers called them puncta. The macrophages were literally full of this glowing tissue protein.
Which immediately suggests the macrophages are consuming the glowing green tissue.
Right. But in science, seeing a macrophage full of green protein isn't enough. You have to prove how it got there.
Absolutely.
Okay, I have to stop you there because my immediate thought upon hearing that is, wait, are we entirely sure they're actually stealing pieces of the cell?
It's a fair question.
Couldn't these green proteins just be leaking out into the surrounding fluid like a burst pipe and the macrophage is just drinking up the puddle? Or, even worse, what if the genetic engineering was slightly off and the immune cells accidentally got the memo to start glowing green themselves?
The researchers knew the scientific community would have those exact same doubts. So they built a series of experimental traps to rule out literally any other explanation.
I love this part.
It's a fantastic narrative of eliminating doubt. First, to prove the macrophages weren't just manufacturing the green protein internally due to some genetic mix-up, they looked at the sheer brightness of the green inside the cells.
Like measuring the wattage of the light bulb.
Effectively, yes. They compared the macrophages from the lung-specific green mice to macrophages from completely normal mice, and then to macrophages from a third group of mice where literally every cell in the body was engineered to glow green.
The ubiquitous green mice.
Exactly. And the intensity inside the macrophages of the lung-specific mice sat perfectly in the middle. It was higher than the normal mice, but significantly lower than the fully glowing mice.
So it wasn't an internal light bulb?
No. That specific middle ground mathematical signature is exactly what you expect when a cell is accumulating a protein from the outside environment over time, rather than turning on its own internal gene.
But they didn't stop with just measuring brightness. The researchers performed bone marrow swaps, which I found totally brilliant.
Oh, the bone marrow chimeras, yeah.
Right. They took normal, non-glowing bone marrow and transplanted it into the green mice.
That is the ultimate proof of origin. The normal donor bone marrow grew into completely normal, non-glowing immune cells. Yet, as those newly minted immune cells patrolled the neon green lungs, they still filled up with tiny green dots.
Incredible.
Yeah, there is no way those immune cells could have manufactured the green protein themselves. They absolutely had to be acquiring it from their environment.
And they ran the experiment in reverse, just to be absolutely certain, right?
They did. They engineered a mouse to glow completely red everywhere. That's the TD tomato mouse. Then they injected a skin cancer tumor, a B16 melanoma, that was engineered to glow green.
Wow.
When they analyzed the environment around the tumor, the host's red immune cells were suddenly full of green tumor vesicles. This microscopic theft wasn't just happening in healthy lungs or healthy skin. It was happening across different tissues and even inside tumors.
And to address my theory about the burst pipe, the idea that the green protein might just be leaking into the fluid, they meticulously mapped the lymphatic system.
Yes, the drainage pathways.
Right, because if a cell dies and simply leaks its contents into the bodily fluids, that protein gets washed downstream into the entire lymphatic network. But when the researchers checked, the non-draining lymph nodes were completely clear.
Exactly. They only found the green dots inside immune cells located in the specific local lymph nodes directly attached to the glowing tissue.
So the macrophage wasn't just drinking from a contaminated river.
Yeah.
It was walking up to the source, taking a piece, and carrying it away.
Yes, very deliberately.
Okay, let's do a quick mental checkpoint here for you, the listener. We are about a third of the way into this deep dive, and so far we've established the central mystery. Macrophages, the immune system's bouncers, are somehow acquiring IDs from completely healthy, living cells. They are bypassing the known pathway of waiting for cells to die.
Right.
We know they have the goods, but we haven't seen the crime yet.
This raises an important question. The tissue they are patrolling, like that slow turnover airway tissue, simply isn't dying fast enough to explain the sheer volume of green protein these macrophages have accumulated inside them.
It's just mathematically impossible.
Exactly. So if the target tissue isn't dead or dying, how exactly are the immune cells extracting it?
Right. How do you catch a microscopic thief in the act when you can't even see the crime scene clearly?
You have to build a much better alarm system. The researchers moved out of the living mice and into a controlled laboratory dish, an in vitro assay, so they could watch the interaction happen live.
So they can actually record it.
Yes, they utilized a highly sophisticated piece of genetic engineering designed to report on the exact vital status of a cell in real time. It's called the B16GC3AI reporter cell.
Wait, so you're saying they essentially built a microscopic traffic light into the target cell? Like red means alive, green means dying.
That is exactly how it functions. It's a dual-color reporter system. As long as the target cell is alive, healthy, and dividing normally, it produces a specific protein that glows red called M-cherry.
Okay, M-cherry is red.
Got it. However, if that cell begins to undergo apoptosis specifically, if it activates an internal enzyme called caspase-3, which is the biological point of no return for cellular suicide.
The point of no return.
Right. When that happens, a switch flips inside the cell stops glowing red and immediately starts glowing bright green because of a GFP reporter.
So the researchers take these reporter cells, which are incredibly healthy, they make growing rapidly with a 99% survival rate in log phase, and they drop the bone marrow-derived macrophages, or BMDMs, into the dish with them. Now, if the textbook scavenging model was entirely true, the macrophages should basically be ignoring the red cells. They should be sitting around waiting for a cell to turn green, indicating it's dying, and then they'd swoop in to eat it.
And the data showed the exact opposite.
Here's where it gets really interesting.
Yeah.
The macrophages took up almost exclusively red material. In fact, 99% of the material the macrophages ingested came from cells that had absolutely no green signal whatsoever.
And to validate the traffic light, they ran a control where they deliberately poisoned a batch of target cells with a harsh chemical to force them into apoptosis.
So they basically forced the traffic light to turn green.
Exactly. In that poisoned dish, the cells turned green, and the macrophages happily ate the green material. But in the healthy, unpoisoned culture, the macrophages were actively taking bites straight out of living, breathing cells, and doing so without triggering the target cell's caspase-3 death sequence.
It's not even really a bite, though, is it? Because when a macrophage normally eats a dead cell-like standard phagocytosis, it engulfs the entire thing. It unhinges its jaw, so to speak, and swallows the cell whole.
Right, a huge meal.
But the sheer volume of red material they measured inside these macrophages was minuscule. It was hundreds of times lower than the total volume of the donor cell. It wasn't a meal. It was just a microscopic sip.
The physical mechanics of how they achieved this non-destructive sip is staggering. And honestly, it explains why this phenomenon went unnoticed for so long in immunology.
Why is that?
It comes down to the limitations of traditional microscopy. Standard high-resolution microscopes blast living cells with intense lasers to get a clear image. Over time, that intense light causes phototoxicity.
Literally burns them.
Exactly. It burns and damages the cells, altering their natural behavior. Furthermore, traditional scanning microscopes are relatively slow at building a three-dimensional image.
Right. If the camera's too slow and the flash keeps blinding the subjects, you're never going to catch a stealthy pickpocket.
Exactly. So the researchers had to use incredibly advanced cutting-edge imaging technology. Specifically, they used NSP-ARC and lattice light sheet microscopy.
How does that work?
Well, instead of blasting the whole sample with a laser, this technology sweeps an ultra-thin sheet of light across the cells. It allows for incredibly sharp three-dimensional resolution at blisteringly fast frame rates, all while keeping the cells perfectly happy and undamaged.
And when they finally got this high-speed 3D camera rolling, what they saw looked less like a bouncer checking an ID and more like a microscopic game of tug-of-war.
It really did.
The technical term the paper uses is trogocytosis-like stretching. To visualize this, I want you to imagine a child pulling a tiny, thin string of taffy off a giant block of candy.
The visual is so striking. The living target cell actually forms a tiny physical protrusion extending outward toward the macrophage.
Like reaching out to it.
Right. The macrophage clamps down on that protrusion and then physically pulls away. You can watch the live cell's membrane and its internal cytoplasm stretching like a rubber band across the gap into the macrophage.
And then snap. The tension breaks, and it separates into a tiny self-contained vesicle inside the macrophage. We are talking about submicrometer volumes, right? Less than 0.05 cubic micrometers.
Tiny. Some of these stolen packages were so small they were almost beyond the physical limits of light to resolve.
Wow.
And the speed of the interaction is the real reason the textbooks missed it. Using the advanced light sheet microscopy, they tracked the entire event. The whole interaction takes less than 10 minutes.
That fast.
Yeah, and the physical stretching and the actual snapping of that taffy, the abscission event where the tiny vesicle pinches off and seals itself, that happens in less than 30 seconds. In some observations, the abscission registered in just 8 seconds.
That is wild. If your standard laboratory camera is only taking a picture every few minutes to avoid frying the cells, you miss the tug of war completely.
You miss the whole thing.
You just look at the screen, and suddenly a macrophage has a new red dot inside it, seemingly out of nowhere. Okay, time for our second quick recap.
Good idea.
We've established the crime, and we have the high-speed security footage. Macrophages are engaging in a highly rapid, contact-dependent process, stretching and snapping off tiny living pieces of our healthy cells without killing them. But that brings us to the most crucial and frankly most fascinating part of this deep dive. Where does the stolen taffy go once it's inside the macrophage?
To understand the fate of that material, we have to look closely at the interior logistics of a macrophage.
Because it's not just an empty bag.
Not at all. A macrophage has highly organized internal routing systems. Usually when a macrophage performs standard phagocytosis, when it swallows a dead cell or a dangerous bacterium, that material is quickly routed to an organelle called the lysosome.
I always picture the lysosome as the cellular incinerator. It is packed with harsh acids and destructive enzymes designed to rapidly melt down and destroy whatever gets dropped into it.
That's a great analogy.
Which makes sense if you just swallowed a live pathogen. You want to melt it down before it breaks out.
But there is a massive logistical problem if the macrophage does that with the live sampled material. If the macrophage throws this tiny, delicate living sample straight into the acid incinerator, the genetic information is destroyed almost instantly.
Right. The bouncer confiscates the ID card, but throws it straight into a shredder before even reading the name on it.
Which defeats the entire purpose of sampling it in the first place, so the researchers needed to map the exact trafficking route of the live sampled taffy compared to the material scavenged from dead cells.
How do you even do that inside a living cell?
They utilized a brand new, highly complex analytical technique called vesicle cytometry. It's a high-dimensional flow-based method for labeling vesicles.
Okay, so think of it like chemically tagging the different rooms in a house to see where the stolen goods are being hidden.
Exactly. They stained early processing compartments with one marker, and they stained the highly acidic, destructive lysosomes with a marker called LMP1.
LMP1. So they can watch in real time whether the red dot goes to a safe room or into the LMP1 incinerator room.
And what's fascinating here is that the high-dimensional data revealed a stark divergence in how the macrophage handles the payload based on how it was acquired. The material scavenged from the dead, poisoned cells, the whole cell meals, trafficked heavily and rapidly straight to the LMP1 positive lysosomes.
It went directly to the incinerator, just as the textbooks predict.
Exactly.
But the live sampled taffy.
The tiny vesicles acquired through that rapid 8-second stretching process actively avoided the incinerator. They were poorly subjected to fusion with lysosomes. Instead, they were routed to distinct, stable compartments within the cell.
So instead of throwing this delicate cellular ID card into the fire, the macrophage puts it in a secure, temperature-controlled glass display case.
That's it.
It keeps the protein intact in a stable depot.
And that stable depot has profound physiological consequences for your immune system. Because the live sampled antigen is protected from immediate destruction, the macrophage can hold on to it and present it to passing T cells over a much longer period of time.
We're talking days, right?
Potentially over several days. And this specific protected trafficking route heavily influences which division of the immune army gets the message.
Okay, let's talk about the different divisions because the paper specifically highlights a differential effect on antigen presentation. Broadly speaking, the immune system has two main types of T cells.
Right.
You have your CD8 T cells, which act kind of like the generals coordinating the overall immune strategy, and your CD8 T cells, which are the highly aggressive assassins that actively seek out and destroy infected or mutated cells.
And the researchers discovered that this specific mechanism, the live sampling, the taffy pull, and the routing to the safe display case, is exceptionally good at priming those CD8 assassin T cells.
And they proved this through chemical manipulation, didn't they?
They did. To prove that the display case was absolutely essential, they ran an experiment where they chemically forced the live sampled material to bypass the safe room and drop straight into the lysosomal incinerator.
Oh.
Yeah. When they artificially forced that rapid destruction, the macrophage's ability to prime and activate those CD8 assassin T cells dropped significantly.
So the bouncer couldn't train the security guards to recognize the VIPs because the ID was burned before the guards could get a good look at it. The staple display case is a mandatory requirement to generate the sustained, long-term signals necessary to keep the immune system educated.
If we connect this to the bigger picture, why does this microscopic tug of war matter for you, the person listening right now, walking around with trillions of macrophages patrolling your tissues? It fundamentally rewrites our understanding of how your immune system maintains its baseline reality.
It means our biological definition of self isn't a static memory. It's an active, continuously updated feed.
Exactly. This continuous, low-volume, non-destructive sampling provides the immune system with a constantly updating, highly stable library of what healthy you currently looks like. It provides what immunologists call tonic signals.
Tonic signals, like a baseline.
Yes. Think of tonic signals as a steady, low-level hum of background information. That hum sustains conventional T-cell populations over time and prevents them from accidentally becoming hyperactive and turning against your own tissues. It's the invisible border control of your immunity, quietly operating in the background, making sure the peace is kept.
But as with all powerful biological mechanisms, the paper points out a terrifying loophole.
It does.
If this is how the immune system learns what is safe, there is a dark side when things go wrong. We know macrophages heavily patrol tumors, and tumors are, fundamentally, living tissue.
The chilling implication here is that cancer cells might be actively exploiting this exact trogocytosis-like process by allowing macrophages to gently nibble on them, and knowing that those tiny sips will be routed to the stable, safe display case pathway, the tumor might be tricking the immune system into developing tolerance.
Oh, that is so insidious. The tumor is counterfeiting a VIP pass and handing it directly to the bouncer, and the bouncer is telling the rest of the club that the tumor is on the guest list.
It opens up an entirely new frontier for pharmacology and cancer therapeutics. If researchers can find a way to medically manipulate that specific vesicular routing pathway
To change the locks on the display case.
Exactly. If we can force the tumor's stolen ID out of the display case and directly into the cellular incinerator, we could potentially strip away the tumor's camouflage. We could break the tolerance and unleash the CD8 assassin cells on the cancer.
It means we have to completely rethink how we approach these diseases by looking at how macrophages sip rather than just focusing on how they eat. And as we wrap up this deep dive, I want to leave you with a final provocative thought to mull over.
Okay.
We've established that this delicate microtheft is a vital balancing act. Right now, as you listen to this, millions of macrophages are constantly gently nibbling on your healthy cells to memorize your identity.
Millions of times a second.
But what happens when that exquisite balance is disturbed by a severe viral infection or chronic inflammation or simply the aging process? Could the very mechanism designed to protect us from autoimmune disease be the hidden trigger for it? What happens if the macrophage suddenly takes a sip that's just a little too large or processes the taffy just a little too aggressively?
It is a profound question. We are talking about a margin of error that is submicrometer in scale, operating in fractions of a second. If that timing or volume is disrupted, the definition of self shatters.
The next time you think about your immune system fighting for you, don't just picture a chaotic battlefield. Picture that meticulous bouncer walking quietly through the crowded club of your body, pulling a single microscopic thread from a patron's jacket, inspecting it in the light, and quietly deciding the fate of the entire system. Thank you for joining us on this deep dive into the invisible world that is constantly reshaping your identity from the inside out. We will see you next time.